Good afternoon. I'm Sudiksha. I'll be talking about the contents of week 6, followed by some questions related to that. Let me just quickly share my screen. Okay. Um, so in the previous weeks, we have learned about classification, evolution, understanding different aspects of the cell, various biomolecules that are present in the cells. Right now, we are looking at various cellular processes that happen and what do they do exactly. Okay. So, one of the important aspects of living cells is the metabolism that goes on within the cell, which requires a lot of components. And this leads to various life activities specific that make a living organism, especially self growth, self renewal, endogenous ability to produce energy, movement of the organism. There are some exceptions to this, such as plants, which are sessile and not motile, or there are sessile bacteria or unicellular organisms, sponges. All of these don't move, but internally there is a lot of processing that is happening. The ability of self replicate or to propagate their genes into their offspring, these are some of the characteristics of a living organism. And with as the living organism grows or as time passes, the living organism needs to adapt to the changes that are happening within their environment. This adaptation can be a short lived adaptation that happens within a generation, or in a Lamarckian sense, it can be a adaptation that other passes on to the subsequent generations. There is a lot of metabolism that goes on within the cells. There are a lot of anabolic and catabolic processes that generate molecules and that degrade molecules. uh, a lot of phenotypic changes that occur within a lifespan of an organism as well. A central property to all of this is the central dogma of life, which tells us how the basic molecules of a cell is made. So, as we know, every cell is made up of proteins, DNA, RNA, etc. These proteins are again made up of amino acids, and there is a every sorry every protein has a unique amino acid sequence arranged in a specific sequence. So note, if you are looking at two very different proteins, there will be a very little similarity between these two proteins. The information to synthesize these proteins is present in the nucleic acids of the cell, specifically in the DNA. This has some exceptions, we'll discuss about that, but the final information that gets copied onto the proteins is present within the DNA of the organism. There is an intermediate stage that happens between DNA and protein known as ribonucleic acids or RNA. So the DNA gives rise to a specific RNA sequence that guide the cellular machinery to synthesize the protein, and this is called as the central dogma of life. Initially, it was initially there was some process. Initially, it was assumed that DNA to RNA, RNA to protein, this flow can be reversed as well, but there have been a lot of experiments that have shown that only some of these processes are possible. For example, DNA to RNA is possible, RNA to protein is possible. However, protein to RNA is not possible. RNA to DNA is possible in a very specific circumstance. DNA can give rise to more DNA and RNA can give rise to more RNA. So the central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information. It states that such information cannot be transferred back to the protein, back from protein to either protein or nucleic acid. So there is no interconversion within the protein, or there is no conversion from protein to RNA or protein to DNA. Okay. The central dogma is the basis of life on earth, and it controls a lot of biological processes. Under normal conditions, the information flows go something like this: sequence dependent synthesis of DNA from pre-existing DNA, which is known as replication, which is essentially copying the information that is present within a strand of DNA to make a new strand with the same information. This particularly happens within the S phase of the cell cycle. Next is the sequence dependent synthesis of RNA from DNA, which is known as transcription. So there are a lot of structural properties of RNA absent in DNA, such as it's very labile, it's very reactive, because of which it is easier to do a lot of downstream processing with the RNA. But because of its instability, it's not a very good idea to store information within RNA. It is very prone to mutations. Because of this, there is DNA which gets converted to RNA, which can undergo a lot of very short processes. So DNA is something that is very that is going to stay throughout the life. RNA gets synthesized again and again, very short lived, can get degraded very fast, and then from the RNA there is a sequence dependent synthesis of protein from, not DNA from pre-existing DNA, rather a sequence dependent synthesis of protein from the RNA, which is a process known as translation. Now the process of replication is controlled mainly by DNA polymerase, and we'll come to that. The process of transcription is controlled by RNA polymerase, and the process of translation is controlled by the ribosomal machinery of the cell. Okay. So, the central dogma of life has three major parts: replication, transcription, and translation. Replication happens within the S phase, where there is duplication of DNA done by utilizing the sequence information of the parent DNA, and the enzyme used is DNA polymerase. Transcription happens within the nucleus, whereas translation can happen in the cytosolic fraction or the nuclear fraction, but mostly happens in the cytosol. The information present in the DNA, that DNA gets converted into information as RNA, which is done by DNA dependent RNA polymerases. So there are different types of RNA polymerases. There are DNA dependent and DNA independent RNA polymerases. But for this course, we'll mostly be looking at DNA dependent RNA polymerases. Translation happens when the RNA that gets shuttled from the nucleus to the cytoplasm is converted into converted as information into proteins in a sequence dependent manner. And there are very specific conditions where other processes might happen, such as conversion of RNA to DNA, which happens via reverse transcription, which does not happen in many organisms. It's mostly organisms like viruses that have the machinery for that. So for like you need DNA polymerase for DNA to DNA conversion, you need reverse transcriptase to convert RNA to DNA. Now the enzyme for enzyme reverse transcriptase is not present within vertebrate systems. It's mostly present in viral systems, because of which these processes, when we do in laboratories, we do it as a synthetic process in our labs. It is not a natural process that occurs. So in cases where the reverse transcription occurs, the uh, nucleic material is RNA. This is very common in viruses which need to mutate very fast. And the so essentially the DNA gets converted to RNA, and then it replicates to multiply itself using the host machinery where the infection has happened. One second. And okay. Uh, so in this case, it's a RNA dependent DNA polymerase. When we are talking about normal replication, it's a DNA dependent DNA polymerase. So the second special circumstances, DNA to RNA, RNA to RNA. Most like in case of reverse transcription, RNA to RNA also occurs in very specific cases in specific viruses. In those viruses, instead of going through a DNA intermediate, the information within the RNA is copied into is duplicated, and that is how the information is propagated. And in this case, it's a RNA dependent RNA polymerase. There are in vitro cell-free systems wherein uh, DNA is directly translated to protein. However, this is not very precisely controlled. It is a very synthetic reaction, and this is not something that happens in normal living scenarios. There are certain infectious proteins known as prions. These are nucleic acid free entities. These have various infectious proteins, and the proteins replicate themselves to create more copies of themselves to infect host organisms. But this is a very unusual pathway and is considered to be an exception to the central dogma. Let's look at DNA replication in slightly more detail. So what is DNA replication? It's essentially duplication or resynthesis of the genomic content that is essential to maintain life within an organism. The DNA has to be replicated very precisely without any mutations. I don't know if you guys know about mutations, but mutations are sequence changes, slight sequence changes that occur within the DNA or RNA sequence. This might be deleterious, or it might not have an effect if it's not some information that is copied onto protein, and if it does not change the polypeptide sequence, it usually does not have much of an effect. However, if it changes the polypeptide sequence significantly, the mutation, then it can be very deleterious. Essentially, the protein won't function properly or might get degraded. So if there is a potential mutation, it might create a changed phenotype. This is a chance event. It might or might not happen, depends on the specificity of the enzymes involved. So there are some points that need to be considered while we look at replication of a DNA fragment. The replication machinery must duplicate the whole fragment. It shouldn't duplicate parts of it. It must be free of errors, or as free of errors as it can be. It must amplify the fragment in a given time. For example, it has to complete within the doubling time of an organism. If it exceeds that, then there is no point, because the organism has to divide after a certain point. Because of this, there might be a chance that the ploidy level or the chromosomal concentration of the organism changes. DNA, some basic recap of DNA. It has 4 nitrogenous bases. A, G are the purines that are present. C and T are the pyrimidines that are present, and there is a repeated hydrogen bonding between these bases. A binds to T and G binds to C, and vice versa. And this linking of the strands is known as hybridization. Uh, the DNA that we find within the nucleus is double stranded in nature. So there is a primary strand, and there is a template strand and a complementary strand, which is known as a coding strand. So usually a lot of processes happen from the 5 prime to 3 prime, and there are specific enzymes that go the other way. Okay, so replication machinery is not only DNA polymerase. There are multiple factors that play into this. Is there a question? Okay. Multiple factors that play into this. The first factor that is involved is primase. This essentially synthesizes a very short nucleotide sequence or a primer at the site of replication to kickstart the replication. Now, in case of prokaryotic systems, there are only a single origin of replication. Especially in case of plasmids, there is a single origin of replication. This is not true for eukaryotic systems. Eukaryotic systems have multiple origin of replication. Otherwise, with the speeds that the enzymes go through, it will take a very long time to replicate the whole DNA sequence. DNA polymerase is the enzyme responsible for synthesis of DNA from the pre-existing DNA fragment. These can't do de novo DNA synthesis, rather they need a pre-existing DNA or RNA primer. There are 3 enzymatic activities of the DNA. There is a 5 prime to 3 prime polymerase property. There is a 5 prime to 3 prime exonuclease property in case there is any issues. There is a 3 prime to 5 prime proofreading property. The proofreading becomes very essential in case the polymerase is very prone to errors. Then the proofreading proofreading machinery comes in, repairs the errors that might have happened during the process of copying the information. In case of E. coli, there are 5 different DNA polymerases, but only 3 are involved in replication. The rest 2 are involved in repair or recombination. So this is in case of E. coli, the polymerases involved are Pol A, Pol B and Pol C with specific effects. So in case of Pol B, there is no 5 prime to 3 prime exonuclease activity. The polymerization rates or the rates at which they elongate the DNA is very different in all of these 3 cases. Okay. The other molecules that are associated with the replication machinery is DNA gyrase, which is required to unwind the double-stranded DNA. So, as you remember, the double-stranded DNA was helical in nature. It needs to straighten out before the copying can start. This is done by DNA gyrase. DNA ligase is required to join the DNA fragments. So, in case of 5 prime to 3 prime, there is a continuous synthesis that occurs. However, in case of 3 prime to 5 prime strand, the replication happens in short strands, and these are later joined on by DNA ligase. There is a single stranded binding protein involved that keeps itself bound to the unwinded DNA so that it does not start rewinding again. Okay. So it is kind of difficult to, because it's the DNA sequence is very long, it is kind of difficult to find the site of occurrence. So there are specific sequences that are required to for the enzymes to locate where to start the DNA replication. So how does it know where to start? Since we know that since we know from previously that A binds to T with two hydrogen bonds, so A binds to T with 2 hydrogen bonds, whereas G binds to C with 3 hydrogen bonds. So if there is a region which is rich in G and C, it is going to be very stable. It's very difficult to break the two strands apart. Whereas if it's a region of A to A, it's a AT rich region, then it is going to facilitate the separation of the strands very easily. So usually the replication machinery recognizes these AT rich regions and then starts binding to that. And this particular region is known as origin of replication. Once. Now once the machinery has identified what the origin site is, it needs to separate out the two strands. And the unzipping at the origin forms a Y-shaped structure known as the replication fork. The replication fork, within the replication fork, different enzymes start assembling and then unwinding the downstream strand so that the replication happens smoothly. There is a short chain of RNA that forms at the 5' end. This is known as an RNA primer here, and it is essential for the DNA. So, because DNA polymerase can't start de novo DNA synthesis. In the elongation step, so first step was that to identify the origin site. Second step is to bind and get the place ready for the copying to happen, which the first step of which is making a short RNA primer. Then comes elongation, where DNA polymerase will start adding nucleotides to the pre-existing short oligomeric nucleotide strand. The incoming deoxynucleotide triphosphate gets joined by hydrogen bonding to the appropriate nitrogen bases as the base pairing rule ATGC, AT or TA or CG or GC. They break their high energy bonds, utilize the energy, and then create a strand. And the energy that is released in this process gets used up in the process of polymerization. So the DNA polymerase can polymerize the deoxyribonucleotides in 5' to 3' direction, which is fine for copying the information from the 5' to 3' direction. You start with the 3' end and start synthesizing. However, the replication fork is not happening here as well. There is only one replication fork. So what happens to the other strand? For the other strand, so this the strand in which there's a continuous synthesis is known as the leading strand. In case of the other strand, it's called as lagging strand. The copying happens in short bursts in 5' to 3' direction. So this is the 5' where the replication fork is, the copying happens, and there are multiple RNA primers in this case. These short sequences are known as Okazaki fragments, and these fragments are later joined to give rise to the lagging strand. The RNA primer gets replaced by DNA molecules, and the gap is joined by the DNA ligase. Then the last step is termination, wherein the replication fork proceeds to the end of DNA or is met with a terminal region which contains multiple copies of the TER sequence. These TERs or termination sequences have the sequence specificity so that they can trap and halt the replication or dismantle the replication machinery. As I said, the DNA polymerase is prone to errors, very less errors, but there is still some chance of errors, but the cell can't afford these errors. If there are lot of errors, then it's going to malfunction and probably die. So there is a proofreading machinery and repair machinery. After every round of nucleotide incorporation, the DNA polymerase has a subunit that runs in the backward direction and checks for accuracy of incorporation. If any error is there, it gets corrected. So, to summarize, there is a first the machinery needs to locate where it starts to where it needs to start the replication. It creates a small RNA primer or a DNA primer. Then the machinery starts copying the DNA. In case of leading strand, the copying happens in a continuous manner, whereas in case of lagging strand, the copying happens in short bursts of few nucleotides. And later, after the copying is done, all of these lagging strand products are the RNA primers are removed from both of these. The lagging strand products are joined using DNA ligase, and you have two strands of the DNA. Next is transcription, which is copying of the information in the DNA into RNA. The RNA that we see can be present in multiple forms. There are other forms there are forms other than these three forms as well, but three primary forms that are very well studied are ribosomal RNA or rRNA, transfer RNA or tRNA, messenger RNA or mRNA. And the synthesis of these RNA molecules from the DNA templates happens by RNA polymerases in a DNA dependent manner. This occurs unidirectionally in a 5' to 3' direction, and the segment which is transcribed from DNA is known as transcription unit. In case of eukaryotes, monocistronic transcription occurs where one coding sequence, one stretch of DNA will give rise to only one polypeptide. Whereas that's not the case for prokaryotes. One particular coding sequence, when read differently, can give rise to multiple polypeptides. A basic structure of the DNA that gets copied to RNA is that the DNA sequence has a promoter region for binding of the RNA polymerase, a coding region, and a terminator region to release the RNA polymerase. The first point is the start site where the transcription starts. So it will start from this start point, synthesize the whole RNA, and then the process stops at the terminator site. Some other terms that are very relevant to all of this is upstream, which is the non-template nucleotide region before the start point. Downstream is so this portion here is upstream. Downstream is towards the 3' end on this side. So during transcription, one of the strands is known as a coding strand or a sense strand, where the other one is known as template or antisense strand. So whatever information gets copied will be from the non-coding strand. The transcription happens based on the non-coding strand. The coding strand is named such because, so if the coding strand has a sequence of ATGC, then the RNA that usually forms will be AUGC. From the RNA that forms from the respective non-coding strand will be AUGC. Okay, so what are the differences between prokaryotic and eukaryotic transcription factor? As we discussed, prokaryotic transcription is polycistronic, whereas eukaryotic is monocistronic. Prokaryotic transcription happens within the cytoplasm because there is no particular nucleolus like structure within prokaryotes, whereas in eukaryotes it happens in nucleus. There is also some transcription that will be happening in mitochondria and such, but which will happen in the cytoplasm, but those have their own machinery to do all of that. Usually prokaryotic transcription is a coupled process. Transcription and translation happens simultaneously, whereas in case of eukaryotic transcription, the transcription happens within the nucleus, then the RNA gets shuttled to the cytoplasm for translation to occur. So it's not a single site process. A single type of RNA polymerase is required for synthesis of all types of RNA in case of prokaryotic transcription, whereas eukaryotic transcription requires 3 different types of RNA polymerase in different forms to synthesize all of the RNA. There is no initiation factor required for transcription, whereas eukaryotic transcription requires some particular transcription factors for initiation. And the RNA polymerases in prokaryotic transcription are simpler in nature. They're only made up of 5 subunits, whereas in case of eukaryotic transcription there are 10 to 15 subunits depending on which polymerase you are looking at. In case of prokaryotic transcription, as I said, there are 5 subunits. 2 alpha subunits, 1 beta dash, and a sigma subunit. The alpha subunits form the core enzyme. Beta and beta dash perform all the catalytic functions, and the sigma subunit recognizes the promoter sequence. So the promoter sequence is the area where the RNA polymerase is going to bind to. The core enzyme is formed by 2 alpha subunits and beta dash, whereas the holoenzyme, the functional enzyme, comes together when there is sigma factor as well. So holoenzyme is the actual functional RNA polymerase. Okay. The prokaryotic promoter consists of a 40 base pair region located near the 5' end of the transcription start site. This consists of 2 6 base pair consensus elements known as print box or Tata box or -35 region, usually Tata box. So it has 10 base pairs upstream of the start point in a consensus sequence of TAT-AAT. And the -35 region has a consensus sequence of TTGACA. So if within a strand of DNA you find these particular sequences, then there is a very good possibility that the that there is a gene that can be transcribed downstream of that. So in case of prokaryotes, the transcription happens in 4 stages: template binding, chain initiation, chain elongation, and termination. The template binding, so as you remember, the DNA is double stranded, tightly wound, so it needs to be opened up so that the RNA polymerase can bind to it. So helicases play a very important role in this stage. So the helicase will unwind the DNA, and usually this is done at a AT rich region so that the unwinding is very fast and does not go back to its previous state. Then the RNA polymerase holoenzyme will bind to this area, to the promoter region. The sigma factor gets released when the chain reaches 10 bases, leaving the core enzyme for further elongation. Just a second. Okay. Then after the binding, then the next step is chain initiation. For chain initiation, so chain initiation is inversely proportional to the melting temperature or the Tm of the strand. If there is a AT rich region, it will have a lower Tm because of double hydrogen bonds. GC rich region will have higher Tm because of triple bonds, and therefore will be more stable. So the efficiency of initiation will be more in a AT rich region. So the initial stage at which the DNA was there is known as closed complex, which gets converted to open complex by melting of the short region of DNA, which unwinds. Then the initiation gets there is a start there is the start of transcription. There is a region in the -10 region upstream of the coding sequence, which is very essential for recognition uh, and for the open state to stay as an open state. So within the RNA polymerase, there are two binding sites for nucleotides. There is initiation sites and there is elongation sites. First, the RNA polymerase needs to bind in a conformation so that the DNA is bound to the initiation site, and as the reaction progresses, the conformation changes so that the elongation site is the one bound to the DNA. So the initiation site binds to the first nucleotide within the open promoter complex at +1 position, which is a purine A or G. It means the first nucleotide will be ATP or GTP. Elongation sites bind to the second incoming nucleotide base pairing at +2 position. The two nucleotides are joined, then the first base is released from initiation site and the initiation is complete. Now you have the open complex, the initiation is complete. Next is chain elongation, which occurs in a 5' to 3' direction. It is carried out by something known as a transcriptional bubble, which forms due to transient separation of the double-stranded DNA into single-stranded DNA. The transcription takes place at the template DNA strand. So this particular thing is known as transcription bubble, where the DNA is separated. There is the there is the transcription machinery present in that bubble. So the RNA chain synthesis basically occurs in 5' to 3' direction by adding nucleotide at the 3' end. The 3' OH group of last nucleotide is combined with the phosphate 5' gamma phosphate nucleotide, and alpha and beta phosphate groups are removed, releasing a pyrophosphate. Creating a phosphodiester bond. In case of bacteria, transcription rate is around 40 to 50 nucleotides per second at 37 degrees, and the translation is also for the same duration, for the same at the same similar rates, because both of these processes are coupled in case of prokaryotes, whereas in eukaryotes this is very fast as compared to prokaryotes. Okay, then RNA polymerase moves along with the DNA and the RNA chain keeps on growing until it reaches the termination site. The the transcription bubble is usually for the length of the hybrid is 8 to 9 base pairs. So the transcription bubble is 12 to 14 base pairs, taking into account some of the surrounding bases. As the RNA polymerase moves, the duplex forms again. The RNA hangs as a free polynucleotide chain. Transcription bubble moves continuously by disrupting the DNA structure, and nucleotides are covalently added to the 3' end of the RNA with the removal of pyrophosphate. Then there is chain termination, when the RNA polymerase stops adding the nucleotide into the RNA chain. This happens when the polymerase reaches the terminator sequence, in which case all hydrogen bonds that hold the RNA and DNA hybrid together break. The RNA chain is separated. The transcription machinery falls apart from the DNA molecule. There are two types of transcription termination in case of bacteria: intrinsic termination, where there is no energy used; rho factor dependent termination, which requires a rho factor molecule. Intrinsic termination happens because of structural changes that occur within the um, RNA molecule that is synthesized. It requires GC rich hairpin structure followed by 7 U residues in such a way that the DNA hybrid forces for RNA DNA hybrid is forced for holding the elongation complex together, and the hybrid gets detached, the elongation complex gets collapses, and because of the hairpin structure, this whole machinery falls apart. This is usually because of complementary base pairing, because of specific residues that are present within the DNA and RNA hybrid. This does not require any energy as such. This happens as a intrinsic process. So there are different steps within intrinsic termination itself. There should be two inverted repeats of GCC present in the DNA template, which gets transcribed, and 6 adenine residues that follow this sequence. So essentially, and this happens for a couple of more times, and essentially the inverted repeats form a hairpin structure that pause the polymerase. This is also called stalling, if I remember correctly. Due to formation of the stem loop, the AU bonds get break down, and this leads to termination, and the RNA molecules get separated, which can then be shuttled outside for translation after some processing. Rho dependent termination requires a Rho protein. It's an ATP dependent helicase that disrupts RNA DNA hybrids. It's an essential protein which causes transcription termination in cases where the sequences do not match the intrinsic termination characters. Rho protein is a hexamer ATP dependent helicase. It contains a RNA binding and a ATP hydrolysis domain. It will first bind to a site upstream of the termination sites, and this particular site is known as a RUT site. So what happens is because of the terminator sequence, the RNA polymerase slows down, and then the Rho protein causes conformational changes within itself to catch the RNA polymerase and detach it from the hybrid. Eukaryotic transcription is different from prokaryotic transcription, requires more factors, enhancers, other transcription factors. So let's look at transcription in eukaryotes. One second. As I said, transcription also happens in the cytosolic fraction within mitochondria and chloroplasts, because they have their uh, own nucleic acid information. So in those cases, the transcription machinery is very similar to that of prokaryotes. However, in case of other eukaryotic transcription, there are three polymerases involved. Pol1 is responsible for synthesis of ribosomal RNA. Pol2 is responsible, or polymerase 2 is responsible for messenger RNA synthesis, and polymerase 3 is responsible for tRNA synthesis. Okay. There was for the prokaryotic sequences, the eukaryotic promoter has some specific sequence which gets recognized by transcription factor. It's usually longer than the prokaryotic promoter because this requires more sequences for more specificity. There is a core promoter which where RNA polymerase gets attached and to form the initiation complex, and there is also a upstream promoter element or UP, which is essentially a GC rich region. So all of the transcription factors bind here, then scan the sequence for the core promoter element where where they start unwinding the DNA. There are other factors that are required for uh, synthesis of the RNA apart from the polymerase. TF2D, which recognizes data box or non-data, which essentially recognizes the core promoter of this TBP or Tata binding protein. There are two types, TBP or Tata binding protein, and TAFs or TBP associated factors. There is TF2A, which is responsible for stabilizing the TF2D. TF2B helps in RNA polymerase 2 and TF2F recruitment. It also helps helps to uh, locate the start site. TF2A helps the RNA polymerase in binding to the promoter. TF2E helps in TF2H recruitment, which is the last step, and TF2H helps in promoter melting with heligase activities and promoter clearance by phosphorylation. So this is the sequence of binding for transcription in eukaryotes. First TF2D gets loaded to the site, followed by TF2A, which opens up space for TF2B, then TF2F, and then TF2E, which recruits TF2H, which can recruit the polymer, which can phosphorylate the polymerase to initiate the transcription. Transcription termination in eukaryotes is also happens in by three different mechanisms. For polymerase 1, it's a row dependent termination. For polymerase 3, it's a row independent termination because of the hairpin loop formation. For polymerase 2, which is for the mRNA, it's more complex and usually coupled with RNA processing event, in which the 3' end of the transcript undergoes cleavage. Is there a question?
Astaga.
Was there a question? So a lot of processing happens when the termination of the transcription occurs. The nascent mRNA or the pre-mRNA usually is processed by attachment of other factors, which can then be utilized for final translation. During the initial phase, when the RNA was starting to get copied, in the 5' end there is a okay, I don't think it's here. In the 5' end there is a methyl cap that gets attached to the 5' end. This methyl cap essentially prevents denaturation of the RNA and prevents the effect of exonucleases on the RNA. To the 3' end, there is a poly A tail that is attached. The transcription of poly A tail followed by cleavage of nascent transcript is such that the poly A tail plus the methyl cap, both of these in coordination prevent the RNA from getting denatured, and also this sequences for some molecules to recognize the entity as an RNA molecule. One more thing that happens is splicing, which which will be in the next lecture. Wait. We went through all of this. Huh. mRNA, there are a lot of post-transcriptional modification happening. There is an addition of methyl cap to the 5' end, a poly A tail to the 3' end, and the intronic regions need to be spliced out. Remember, the prokaryotic system does not have introns, so there is no reason for splicing in the prokaryotic system. Also, in case of prokaryotic system, all of the processes is happening in a coupled manner. So after the transcription, the molecule immediately goes for translation. So there is no need for it to have a stabilizer of any sort. Capping in eukaryotic cells, mRNA is inherently unstable at the ends because of ribonucleases. So the mRNA is capped so that it is protected by ribonucleases, and it can bind to the ribosome for translation. The capping reaction occurs as soon as transcription has started. Around 20 to 30 nucleotides are formed and the capping occurs. This is the reaction that it follows, in which 7-methylguanosine attaches to gives its methyl group for methyl capping. At the 5' end, capping process occurs. A slightly modified guanine, 7-methylguanosine or 7-mg, is attached backwards by a 5' to 5' linkage to the triphosphates of the first transcribed group. This is a condensation reaction in which the methylguanosine, the methylation happens at the guanine at N7, and there are further methylations occurring at the 2' hydroxyls of second and third nucleotides to the adjacent cap. There is one more step, tailing or addition of a poly A tail, wherein a series of adenosine residues ranging from 80 to 250 forming a poly A adenosine tail at the 3' end of the primary transcript is done. It is, as I said, it can there are certain molecules that will recognize these 5' and poly A tail sequences to know that the entity is an RNA molecule. So the poly A tail can be used to export mRNA out of the nucleus. It increases the stability of the mRNA, and it serves as a recognition signal for binding of translational factors during the initiation of the translation. This process requires a template independent RNA polymerase known as poly A polymerase. Then comes splicing. Eukaryotic systems have intronic and exonic regions within the DNA, and all of this information gets copied. But the intronic regions are not required for the protein function. If the intronic regions are included in the translation process, then it creates a very different protein, and this protein may or may not have the function of the initial protein. So, the intronic sequences essentially do not appear in the final mRNA. The length of the introns can vary. There are it can have one particular gene can have more introns than exons. It can have multiple introns, and depending on what gets included and what gets excluded, very different protein structures get formed. So there are 4 classes of introns, group 1 and group 2. Both of these are self-splicing introns and does not require any protein enzymes. Spliceosomal introns, they are not self-splicing, they require a machinery to splice themselves. Introns that require ATP for splicing is the 4th category. So group 1 and in case of group 1 and group 2, the splicing mechanism involves a trans-esterification reaction in which a ribose 2' or 3' hydroxyl group takes an nucleophilic attack on the phosphorus and forms a new phosphodiester bond on expense of old one, thus excluding the intronic regions. There are slight differences between these two. In case of group 1, the guanine nucleotide is the nucleophile, whereas in case of group 2, it's the adenosine where the formation happens. And there are other spliceosomal machinery present beyond the scope of this course, but in the end, the all the introns get removed, and some or all of the exons are kept within the mRNA. Now there can be inclusion of different exons. uh, it is not essential that all of the exons are kept within the mRNA, and this process in which multiple products can form from a single mRNA is known as alternative splicing. In this, only some of the exons are kept and some are excluded. This is essentially done to create splice variants, and these splice variants can have proteins of very similar function or very different function depending on their structural uh, properties. One of the examples is occurs in case of sex determination in Drosophila, in which sex lethal transformer gene and double sex gene, all of these are uh, all of these can create different splice variants. Due to alternative splicing, functional genes are produced in females and non-functional genes are produced in males, and because of this, there is gender assigning in case of Drosophila. This also happens in case of immunoglobulins, in wherein there is a variable domain present. So some exons are kept and some are not kept, creating a very different immunoglobulin. This also happens in case of hormones, wherein a calcium regulating hormone and the calcitonin gene related peptide are produced due to the splicing event from a single gene. Okay. So up until now, the methyl capping, the poly A addition, and the splicing occurs in case of messenger RNA. That is not true for ribosomal RNA. Ribosomal RNA have different processing. So in case of eukaryotes, the ribosomal RNA is 80S, whereas prokaryotes have a 70S ribosome. Um, they are transcribed, the ribosomal RNA are transcribed as a long precursor sequence, which are then modified at specific bases and cleaved to give more mature products. So so the two post processing that happens to ribosomal RNA is cleavage and base modification. Okay. So in case of the 30S RNA, it's modified and cleaved to give 23S RNA, 16S rRNA, 5S, and some tRNA segments. So, and similarly, in case there might be some intermediate products that form, but these from a single pre-rRNA, all of these products can form. In case of eukaryotes, a single rRNA can give rise to a 90S pre-ribosomal nucleolar complex can give rise to 18S, 5.8S, and 28S rRNA. The cleavage and modification is guided by SNO RNAs or small nucleolar RNAs. Remember that I said that mRNA, tRNA, and rRNA are not the only RNA molecules that are present. SNO RNA or small nuclear RNA is one such other RNA molecules present. There are long non-coding RNA, there are micro RNAs present within the cell that also affect various cellular processes. In case of yeast, there is a pre-RNA that gives rise to 170 non-ribosomal proteins, 70 SNO RNA, and 78 ribosomal proteins. So essentially, SNO RNA are somewhat related to spliceosomes. In case of both eukaryotes and prokaryotes, tRNA processing occurs. This is transcribed as a long precursor with a single primary transcript that carry more than one tRNA segments, which are separated by cleavage. So for in case of tRNA, it's not only the cleavage for separating the tRNA segments, the tRNA also need to capture the amino acid for which it is responsible for. So the processing of pre-tRNA involves cutting off of extra sequences by endonucleases as RNA at the 5' site and RNSP at the 5' site and RNSD at the 3' site. Was there a question? After removal of the sequences from the 3' end, CCA sequence is added by a enzyme tRNA nucleotide transferase here. Then the enzyme binds to the CCA sequence at its active site and phosphodiester bond is formed with the 3' end. Furthermore, there is a base modification occurring simultaneously as methylation, determination or reduction. In case of pseudouridine, uracil uracil is removed and attached to the sugar to the C5, which happens here. Now let's look at translation. Which is required for making the protein molecules from the information present in the RNA. Okay. So, the messenger RNA has a 5' end, a 5' untranslated region, which is not necessarily translated. This also contains the methyl cap, the ribosomal binding site, and the coding sequence, and the 3' UTR. In eukaryotes, there is a poly-A tail as well. So, methyl cap, 5' untranslated region, start site, stop site, which between which the coding sequence is present, 3' UTR, and the poly-A tail. So, the messenger RNA has 3 reading frames, out of which only one codes for the desired protein. If in the sequence of bases there is no stop codon to interrupt the translation, then the synthesis of the entire polypeptides chain occurs, and this is known as open reading frames. So, suppose you have A, T, G, C, T, A. Okay. This is just an example. It will be substantially longer from this, but the start can happen here from ATG. So the translation machinery recognizes the RNA sequence in form of three nucleotide sequences, which is known as a codon. So the translation can start from here, in which it codes for 2 amino acids. It can start from here, in which case it codes for 1 amino acid, or it can start from here, in which case it codes for this, codes for a this won't be T, this will be U. AUG, CUA, if this is the case, then depending on where it is starting to read, this can be one amino acid and this can be the other. This can be one amino acid or this can be one amino acid. So depends on where it's starting. So the mRNA is a random sequence of nucleotides differentiated by the bases attached to them, which are uracil, adenine, cytosine, and guanine. And these when considered in three nucleotides together, code for specific amino acids, and these 3 are called as codons. So why 3 in particular? Why not 1 or why not 2? If it was 1, you can only have 4 amino acids. If it was 2, you can have 2 to the power 2, 4 to the power 2, 16 amino acids. But in reality, there are 20 amino acids with some exceptions. These 20 amino acids, because there are 20 amino acids, the codon needs to be in triplet. So 2 to the power 3 gives 64 possible amino acids. There are 20, so there is some redundancy or degeneracy between different sequences. And this forms the basis for Wobble hypothesis, wherein multiple, depending on the first two nucleotides, the third there is some leniency in the third nucleotide. So the first two are very essential for what the amino acid is going to be, and the third one has specificity but not so much. Okay. The genetic code is a triplet code known as codon. Each coding sequence has a start and stop codon. The start codon is usually methionine, which is AUG. The stop codons are UAA, UAG, and UGA. So these three are the stop codons. This is the start codon. The code is ambiguous, which suggests that the code is only for one amino acid. There is no comma, gap in the code, and the code is degenerate, which means one amino acid has more than one codon. For example, phenylalanine has UUU and UUC. Leucine can be coded by UUA and UUG. There are only very specific amino acids which are coded by one. Examples are methionine, which is coded by AUG, or tryptophan, which is coded by UGG. There are some amino acids which are coded by as much as 4 or 6 codons. One example is leucine, which is coded by 6. The codon is non-overlapping. For example, if this is the sequence, then this is your start site AUG, which codes for methionine, then CUG, then GGU, GAU, etc. It won't happen that it is AUG, then UGC, then GCU. That is not going to be the case. So if you were to translate this will be methionine, then CUG will be lysine, then GGU will be GGU will be glycine, and you can make the whole sequence by using this table. It is universal, which suggests that the genetic code and its meaning is common for all life forms. There are some exceptions. For example, UGA is a stop codon in case of vertebrates, but it codes for tryptophan in mycoplasma, spiroplasma, and mitochondria in several species. So there is some discrepancy between the codons when you are comparing it between eukaryotes and prokaryotes, but within the eukaryotic system it's usually consistent. Okay. The transfer RNA is very essential for translation. This has a clover leaf structure wherein there are 3 circular arms. There is a D arm, there is an anticodon arm, there is a T sigma C arm. The sigma codes for pseudouridine, which is a modified nucleotide. There is the amino acid arm, and there is the anticodon. The anticodon arm contains the anticodon sequence, which is going to recognize the codon sequence. The amino acid arm catches the different amino acids and depending on the anticodon, and which can be added during the translation process. So there is a 5'-phosphate terminal here, an acceptor arm that ends in CCA terminal at 3'. Here D loop, which contains dihydrouridine, anticodon loop, T arm which has T sigma C, which is where sigma is the pseudouridine. CCA sequence is important for recognition of tRNA and also is the site of attachment. So the amino acid attaches somewhere here. Each tRNA is specific to amino acid and that it carries CCA in its arm. There are 30 to 45 different tRNA in prokaryotes and 15 eukaryotes, which suggests that there is more than one tRNA for a single amino acid. For example, for glycine, there are 2 tRNA, gly1 and gly2. So, the anticodon is going to recognize the codon. It will have the appropriate amino acid here, and then by repeating this process, you make the whole protein. The translation happens within ribosomal machinery in the cell. The ribosomes are ribonucleoprotein particles that contain rRNA and protein molecules. So it's a complex. Each ribosome is made up of two subunits, a 50S and a 30S subunit in case of prokaryotes. In case of E. coli, the 30S subunit consists of 16S rRNA, 21R proteins or ribosomal proteins. The 50S subunit consists of 23S rRNA, 5S rRNA and 31 proteins. In case of mammalian, eukaryotic system, the setup is a 80S ribosome, which contains 60S and 40S ribosomal proteins. This is the big subunit, this is the small subunit. The 60S subunit consists of 28S rRNA, 5S rRNA, 5.8S rRNA and 50 ribosomal proteins. 40S subunit consists of 18S rRNA and 33R proteins. So the 70S ribosome has 3 tRNA binding site, a P site or a peptidyl tRNA binding site, a site amino acyl tRNA binding site, or a E site or a deacetylated tRNA, or it is also called as the exit site. So first, if we look at the mechanism of translation, the amino acid needs to be ready for addition into the polypeptide chain. So there is activation of amino acids and tRNAs, in which the amino acids are attached to tRNA in presence of an enzyme called amino acyl tRNA synthetase, which occurs in presence of amino acids tRNA with energy expenditure as in terms of ATP. So the amino acid, in presence of amino acyl tRNA synthetase, it will form an amino acyl tRNA complex with a release of a pyrophosphate group and AMP molecule. This enzyme activates amino acids by attaching covalently to the tRNA when tRNA gets charged and is named as amino acyl tRNA. This is a very high energy bond, and these tRNAs are known as activated amino acids. In case of eubacteria, first amino acid in the polypeptide chain is N-formyl methionine, which is specific to AUG, GUG and UUG. Whenever these codes are present, it's the initiation point where there will be an N-formyl methionine, but if these sequences are present between the coding sequence after the first initiation, then they code for methionine and valine respectively. So there is the initiator tRNA is very different from the rest of the tRNA, and it has unique features that enables the initiation of the translation. So in the first step, the small subunit of ribosome binds to the mRNA such that the initiation codes lies in the partial P site. So it lies somewhere in between here, A and P site. So the entry of the RNA always occurs from this end. It first and comes in contact with A, then P, then exits from E. So this happens by initiation factor 3. It prevents untimely reassociation of the large and small subunit of the ribosome and promises the accuracy of initiation site selection. Within the mRNA, there is a ribosomal binding site called as the Shine-Dalgarno sequence and the initiation codon. The Shine-Dalgarno sequence is 3 to 8 nucleotides ahead upstream of the initiation codon. There is a specific sequence AGG-AGG-U located around 10 base pair of the initiation codon, which this particular sequence is complementary to a region near the 3' of the 16S rRNA, which is a component of the small subunit of the RNA. So, this enables the recruitment of the initiation factors, the complex forms, the ribosomal complex forms. The next step, initiator tRNA carrying N-formyl methionine enters the partial P site and binds to the mRNA via the anticodon loop. At this point, IF2 enters the picture. It directs initiator tRNA to the correct position in the initiation complex. It also exhibits ribosome dependent GTPase activity, and once the GTP is hydrolyzed, the 50S subunits attaches itself to form the whole complex. When the complex is complete, it forms a complete A, complete P and a complete E site. So between prokaryotes and eukaryotes, there are some major differences. The first difference is in the initiation process, wherein in eukaryotes there is only one start codon, AUG, whereas in case of prokaryotes there is 3 start codons. This AUG in eukaryotes does not code for N-formyl methionine, rather it codes for methionine. So eukaryotic cells need more initiation factors to regulate this process. There are 12 initiation factors in case of eukaryotic cells. There are 3 in case of prokaryotic cells. In eukaryotes, the process of association of mRNA with the smaller subunit is more complex than prokaryotes. The 40S subunit first identifies the 5' methylated cap, and then there is a scanning process involved wherein initiation codon is recognized. Once this recognition is aided by ATP dependent helicases that hydrolyzes the ATP, and this is the initiation codon recognition occurs by Kozak sequence, similar to Shine-Dalgarno sequence in case of prokaryotes. Okay. So only once this recognition and everything is done, only then the elongation starts. It's a cyclic process. It starts from the formation of first peptide bond to the addition of last amino acid. The amino acid is added to the chain one at a time. Addition of the amino acid is a very rapid process, otherwise it will take forever to make the protein. The peptide sequence is in the order of codon and anticodons in the mRNA, and the rate of elongation is around 15 amino acids per second, so a very fast reaction. This is catalyzed by multiple enzymes, so very fast. So what is required for the elongation to start? The mRNA and the 70S or 80S ribosome is present, the amino acid tRNA is present, the elongation factors are present, the initiation has happened. These are very essential, only then the elongation starts. There are multiple elongation factors as well. EFTU is a G protein that binds to amino acid tRNA and directs correct position at the ribosome site A. EFTs regenerate the EFTU and by hydrolysis of GTP. EFG is also a G protein which mediates translocation. Is there a question? The elongation is carried out by the ribosome in 3 different stages. First, it needs to read the information present within the mRNA and bring in the appropriate tRNA. So, the ribosome select and bind to the incoming amino acid tRNA at a site whose anticodon is complementary to the codon of the mRNA. decoding the region of 16S rRNA to confirm the proper base pairing between codon and anticodon. Next, the peptidyl group of the P site of the tRNA is transferred to the amino acid group in a site through the peptide bond. Next, the tRNA in the A site is transferred to the P site to make a space for next amino acid tRNA at A site. And the one present in P site is, this would be P. P site is shifted to E site. This is also coupled with ribosome movement along with mRNA. So it's a antiparallel. It will be happening in the antiparallel direction. The process of chain elongation in the ribosome is mediated by multiple elongation factors, as we saw. EFTU promotes the entry of amino acid tRNA to the A site of the 70S ribosome. Then the EFTU binds to a GTP molecule, making a binary complex, which creates an activated EFTU complex that can then bind to the tRNA to make a ternary complex. When the codon-anticodon base pairing is stabilized and hydrolysis of GTP occurs, it converts by conversion of GTP to GTP plus PI. It helps the ternary complex to bind to the A site, and after this whole complex gets released. Then the complex can bind to another GTP molecule and repeat the same process again. In case of peptidyl transfer, the amino acid present at the P site has a COO minus or a COOH group present. This COOH will react to the amino acid present in the A site and will make a CONH2 group, a peptide bond, using in presence of a peptidyl transferase enzyme, which is present in the ribosomal subunit. This enzyme is associated with to the 23S rRNA of the 50S ribosomal subunit and creates an amide bond which mediates the elongation of the peptide, of the oligopeptide or a polypeptide. Next step is translocation, in which 3 steps are necessary. The deacylated RNA needs to be removed from the P site. The peptidyl tRNA needs to move from A to P site, and the ribosome should move on the mRNA one codon down, so the next codon can come at the A site. This is carried out by EFG factor, in which the tRNA of A and P sites interact with the peptidyl transferase center, make a polypeptide bond, and then the deacylated tRNA and this gets released. The deacylated tRNA then moves to the E site, and the newly formed peptidyl tRNA moves from A to P site. Then there is the termination. The termination of translation occurs due to the stop codon. You saw there were 3 stop codons, UAA, UAG and UGA. If any one of these appears at the A site of the ribosome, then termination occurs, because none of the tRNAs have anticodons that recognize these codons. So therefore there is termination of the reaction, and during this process termination release factors are involved, which bind to the ribosomal machinery. RF1 binds to the ribosome when UA or UGA is in A site. RF2 binds to the ribosome. RF3's main function is to catalyze the release process through GTP binding and hydrolysis. So for the termination process, RF1 or RF2 binds to the ribosome at the near the A site. Polypeptide chain is released from the ribosome by the peptidyl transferase complex, and the peptidyl transferase complex transfers the carboxy terminal residue of the polypeptide chain from the tRNA of P site to the water molecule. The RF molecules and GDP get released. tRNA is also free. But because of release of all of this, the 70S ribosome is unstable due to presence of initiation factors, and the ribosome dismantles, recycling happens. It gets prepared for the next cell. So whatever we get from here is a polypeptide sequence in which only amino acids are present. Then there are a lot of post-translational modifications happen to this protein sequence, like for the mRNA sequence. These are known as post-translational modifications, or PTMs, and these have a very big role in various signaling molecules. One of the major post-translational modification is phosphorylation. It is prevalent in all of the organisms. It alters the catalytic activity of proteins. So phosphorylated versus a deep phosphorylated protein have very different properties. They might have conformation, there might be conformation changes due to the phosphorylation, or it might change the activity of the protein. A phosphorylated form might be more active than a non-phosphorylated form. This phosphorylation might be a very long-lived process or might be a short-lived process. Mostly phosphorylation acts as a switch between an active and inactive form to enable the signaling pathway. So if the signaling pathway is going in a certain way, if everything is active, then the protein might get phosphorylated. If this is the protein, then the protein might get phosphorylated. However, if the phosphorylation is not there, the protein won't function and none of the downstream processes will happen. So it functionally regulates the catalytic activity of protein by defining a rigid permanent 3D protein structure. Temporary phosphorylated proteins serve as anchor for other protein substrates in signal transduction pathways. It regulates many cellular processes like cell cycle, cell growth, apoptosis, reduction of signal transduction pathways. In eukaryotic cells, phosphorylation occurs in the side chains of 3 amino acids: serine, threonine, and tyrosine. Usually, these are the 3 amino acids in which we see phosphorylation, and there are specific antibodies that recognize these phosphorylation as well. So, if you want to see an active versus an inactive protein, we can selectively stain with phosphorylated antibodies to see what the status of the protein is in a cell. So the amino acids harbor a nucleophilic OH group. The terminal phosphate on the universal phosphoryl donor ATP, or it also might happen from GTP in certain cases, serves as a point of nucleophilic attack from the OH group, which results in transfer of phosphate groups to the amino acid side chain. Magnesium ions act as catalyst by chelating the gamma and beta phosphate groups, resulting in lowering of the threshold for the phosphoryl transfer to the nucleophilic group. The phosphorylation, as I said, can lead to conformational changes. The conformational change stimulate the catalytic activity of the protein, so the protein can be activated or inactivated by the phosphorylation. Phosphorylated proteins employ neighboring proteins which have structurally conserved domain to distinguish between the phosphorylated motifs, and these domains are specific for diverse amino acids. So there is a specific sequence of polypeptides that will get phosphorylated, and a lot of proteins recognize these motifs and do a lot of downstream signaling. This usually is a reversible post-translational modification, so a lot of recycling happens. The protein might shuffle between a phosphorylated and an unphosphorylated state, or a dephosphorylated state. Two types of enzymes make it possible for the dynamic nature of phosphorylated proteins: kinases and phosphatases. So kinases add the phosphorylate phosphor pi group to the phosphate group to the protein, whereas phosphatases remove the phosphate group. Okay. Another post-translational modification that occurs is glycosylation, which happens, if you remember the Golgi ER machinery, this happens in the function between the ER and Golgi apparatus. So with the rough endoplasmic reticulum, a lot of the proteins get synthesized, then they shuttle into the Golgi for post processing. During this post processing, a lot of proteins, especially the proteins that are present at the surface of the cell, to these proteins glucose or sugar molecules are added. Approximately 50% proteins characteristically expressed in a cell go through this alteration, which involves covalent addition of sugar moieties to the specific amino acid. This process may or may not be reversible. Usually it's an irreversible process, but if there is a reversal needed, a lot of molecules are there, a lot of enzymes play a role in this. Mostly soluble and membrane-bound proteins expressed in the endoplasmic reticulum undergo glycosylation, including all secreted proteins, surface receptors, and ligands. Some proteins that are transferred from Golgi to the cytoplasm are also glycosylated. Ubiquitination is another post-translational modification in which ubiquitin molecule is added to the protein. It's a eukaryotic protein. The ubiquitin is a eukaryotic protein coded in 4 different genes in mammals. These are the 4 different genes. This UBA52, RPS27, AUBB, UBC. Sorry, RPS27A, UBB, and UBC. Proteins are made up of 76 amino acids. Molecular mass, these have around 8.5 kDa molecular mass. This is characterized by presence of a C-terminal tail and 7 lysine residues. The carboxylic acid of the terminal glycine from a diglycine motif in an activated ubiquitin forms a amide bond with the epsilon amine of lysine of a modified protein. So this happens to the protein should have a lysine, and the ubiquitinylation occurs on this lysine molecule. This, in most cases, in a lot of cases, the cellular protein, if ubiquitinated, undergoes degradation by proteasome. So ubiquitination is a signal for the proteasomal machinery to degrade the protein. It can be responsible for change in the location of the protein from one subcompartment in the cell to another subcompartment, or it can mask certain interaction sites on the protein to prevent protein-protein interaction. Activation of ubiquitin occurs as a two-step reaction. Ubiquitin interacts with ATP, forms ubiquitin adenylate intermediate. In the next step, ubiquitin is transferred to the E1 active site containing a cysteine residue. This causes formation of the thioester linkage between the C1 carboxyl group of ubiquitin and E1 cysteine of sulfur group. And then in the last step, there is an isopeptide bond between lysine of the target protein and the C-terminal of the ubiquitin via activity of one of the 100 E3 ubiquitin protein ligases. So in this cascade, one UN molecule causes binding to several E2, which in turn causes binding to hundreds of E3 in a hierarchical fashion. Another post-translation modification is methylation, in which the methyl group can be added to the nitrogen N-methylation or oxygen O-methylation of the amino acid chain. N-methylation is irreversible, whereas O-methylation is potentially reversible, and therefore O-methylation might be a switch like phosphorylation, whereas N-methylation leads to formation of more unstable or stable products. Methylation enhances the hydrophobicity of amino acids, neutralizes negative charge attached to the carboxylic acid. Main methyl group contributor is SAM or S-adenosylmethionine. This also is true for the mRNA processing. SAM is also the major methyl contributor in that case. This reaction is mediated by enzyme methyl transferases, and this methylation is important for a lot of epigenetic regulations, such as histone methylation and demethylation. So depending on the methylation status, the histones might be arranged in a more compact or a more dispersed fashion. Depending on which residues the methylation happens, the chromatin shifts between eukaryotic to heterochromatic moieties. Okay, let's discuss some questions pertaining to this week's contents. Let me just share my screen. Okay. Unless there is a question. Is there a question? So the first question is, the cellular compartment where transcription occurs in eukaryotes is cytoplasm, nucleus, Golgi complex, or endoplasmic reticulum? The answer is nucleus. So replication will happen within nucleus or within mitochondria or within chloroplast, but mostly within the nucleus. No, it's B. Transcription will happen within the nucleus. Translation will happen in the cytoplasm, and there will be post processing, after which the translated products can go wherever they want. The property that makes Taq polymerase ideal for PCR is that it's derived from eukaryotic cells. It can function at high temperatures without denaturing. It is specific to RNA templates, or it requires low temperature. It is stable at high temperatures. I don't think we discussed PCR, but in PCR the steps are denaturation, annealing of the primers, and elongation. So the denaturation occurs at very high temperatures. Because of this, if the enzyme, the polymerase, is not stable at high temperatures, it won't be able to do its polymerase action. Primers are required in PCR to degrade the DNA template, to prevent contamination, to stop DNA replication, or to provide a free 3' hydroxyl group for DNA polymerase to start synthesis. The answer is D. To provide a free 3' hydroxyl group for DNA polymerase to start synthesis. Function of a spliceosome is initiation of transcription, adding a poly-A tail to RNA, removing introns and splicing exons together in pre-mRNA, or terminating transcription. It's okay, answer. It's fine. The answer is C. The regulatory sequence that is necessary for binding of transcription factors is promoter region, exon, splice site, polyadenylation signal. It's the promoter region. GTP, function of GTP during translation is to catalyze peptide formation, provide energy from movement of ribosome along mRNA, bind amino acids to tRNA, or terminate translation. The answer is to provide energy for movement of ribosome along mRNA. Ribosome recycles after termination of translation by attaching to another mRNA molecule immediately, undergoing enzymatic degradation, dissociating into small and large subunits with the help of release factors, or binding to the same mRNA again. The answer is dissociating into small and large subunits with the help of release factors, following which, if needed, it can reassemble again, attach to another mRNA molecule or the same mRNA molecule, and translate the information. Correct matching of codons and anticodons during translation is ensured by the ribosome's A site, the enzymatic activity of rRNA, complementary base pairing between codons and anticodons, or presence of elongation factors. The answer is complementary base pairing between codons and anticodons. The enzyme that links amino acids during protein synthesis is DNA polymerase, peptidyl transferase, RNA polymerase, ligase. It's peptidyl transferase. It catalyzes the formation of the peptide bond. Termination of translation is signaled by a release factor binding to stop codon, end of mRNA sequence, addition of a poly-A tail, tRNA degradation. The answer is a release factor binding to a stop codon. Poly-A tail in eukaryotic mRNA initiates DNA replication, directs the RNA to spliceosomes, signals transcription termination, protects RNA from degradation, and aids in translation. The answer is protects RNA from degradation and aids in translation. The role general transcription factors play in eukaryotic transcription is enhancing the degradation of mRNA, assisting RNA polymerase in binding to promoter, catalyzing the splicing of pre-mRNA, directing the translation of mRNA. Answer is assisting RNA polymerase to binding of the protein. Fidelity of RNA replication is maintained by helicase action, proofreading activity of DNA polymerase, continuous unwinding of DNA helix, the synthesis of RNA primers. The answer is proofreading activity of DNA polymerase. Feature of replication origins that ensure accurate DNA replication is by high GC content, binding sites for topoisomerase, presence of multiple adenine thymine rich sequences for easy strand separation, or RNA primers bound permanently. The answer is presence of multiple adenine thymine rich sequences for easy strand separation. Function of topoisomerase during replication is relieving supercoiling stress ahead of the replication fork, joining Okazaki fragments, adding nucleotides to the growing strand, or initiating replication. The answer is relieving supercoiling stress ahead of the replication fork so that the replication happens smoothly. Are there any questions? Please ask if there are any doubts or something wasn't clear. If there are no further questions, this is the end of the session. Thank you for listening. Thank you.